Hey everyone, welcome back to part 2 of the alpha-complementation blue-white screening videos. In this video, I'll be talking about how blue-white screening works and why researchers use this. If you're watching this video first, I'd recommend you actually take a look at part 1 before watching this video, because part 1 addresses the concepts of alpha complementation, which is the foundation behind blue-white screening. While blue-white screening is really pretty easy to understand, I'm going to be using some analogies and relating them back to the science to help illustrate how this all works. So if you found yourself having a bit of a hard time understanding it all, then you're in the right place. So for blue-white screening, I'll be using a different analogy, different from the one that I used in part 1. Imagine you need a bookshelf. So you go to the store and you buy one of those nice ones where it's the bookshelf that stacks on top of the cabinet base. And let's also imagine that you have a really wonderful friend who has agreed to help you build this thing. To make the job go faster, you decide to work on the cabinet portion while your friend assembles the bookcase part. And once you're finished with each of your sections, you'll stack the bookcase on top of the cabinet and you're done. Fortunately, in this analogy, we'll say too that the manufacturers anticipated this teamwork and crafted their instructions into 2 sections, a section of instructions for the cabinet and a section of instructions for the shelving part. But as you start to pull everything out of the box and get organized, another friend comes over. And this friend is somewhat of a prankster. And the prankster decides to have a little fun with you guys. So he picks up your instructions for the cabinet part and decides to add in a few lines of his own instructions within the manufacturer's steps. Now, when you follow this set of instructions with both the manufacturer's steps and the prankster's steps, the cabinet that you're trying to build won't function. In fact, if you want to get technical, what you build isn't even a cabinet at all. And when you and your friend are finished with each of your section, you really can't stack them together. This is similar to how scientists use blue-white screening and alpha complementation. And they use it to confirm whether their gene of interest was taken up or not. Another way of looking at it is that the researcher inserting the genes into the plasmid is much like the prankster who inserted the instructions into part of the manual. But since we're working on a molecular level, the only way for researchers to see whether their instructions were actually added is to see what happens in the end, and blue-white screening gives us that visual element. Before we go deeper on this, I just kind of want to map the players of our bookcase analogy to the blue-white screening process. You, the person building the cabinet, would represent the plasmid with the alpha peptide instructions. See video part 1 on that one. Your friend who is helping build the shelving section would be like our E. coli cloning cell, and their set of instructions would be the cloning strain's genome that carries the sequence for the omega peptide, the peptide with a missing piece in it. When you and your friend put each of your sections together, it's like the missing piece is uniting with its other part. Then there's the prankster, and this would be similar to the researcher who's cloning new genes into the plasmid. And the prankster's instruction that they inserted into the manual would be the desired genetic sequence a researcher is trying to insert into the plasmid. When the researcher inserts their instructions into the plasmid, they do it in a way that disrupts the sequence for the alpha peptide. And when that happens, the alpha peptide won't actually get produced. Again, very much like our prankster who wrote in their own steps in the middle of the manufacturer's instructions. Okay, so now that we have the who's who, we can talk more about the principles of blue-white screening. In blue-white screening, blue colonies form on the plate when a functional omega peptide combines with an alpha peptide, allowing that tetramerization. This is because of a blue precipitate that occurs when the functioning beta-galactosidase in E. coli breaks down xgal. But if the gene for the alpha peptide is disrupted by the insertion of a researcher's target gene, the alpha peptide isn't produced and complementation doesn't happen. This would be visually shown with white colonies growing on the plate since there's no functional beta-galactosidase enzyme breaking down xgal. Now let's put it all together. Researchers are using blue-white screening to see if their plasmid actually took up the target sequence or not. And we know that when it comes to blue-white screening, we can visually confirm if the gene was inserted because the inserted gene will disrupt the sequence that produces the alpha peptide. In colonies where this happens, this leads to no no alpha complementation, and those colonies would appear white. In practice, some cells will be white where gene insertion was successful, and others will not have been successful and appear blue on the plate. From there, researchers can select colonies that actually carry their target gene for further work. And that's really it. That's the long-winded way of understanding the concepts behind blue-white screening. If you're interested in learning more, we also have a very brief but very helpful video. It's a whiteboard presentation about alpha complementation, and I'd encourage you to take a look at that one too. Between that video and these two videos and putting it all together, you should have a decent understanding of the whole process. Be sure also to check out goldbio.com for other helpful resources. And if you're in the market for affordable, high quality reagents, browse our site. Thanks, everyone, and until next time.